It's a pleasure to introduce my co-organizer, Dr. Shakti Menon. So Shakti did his PhD from University of Sydney and went on to do a postdoc at Brisbane, Queensland University of Technology. And incidentally, he worked on wound healing, which actually has been referred to multiple times in this workshop as being very close to dorsal closure. So he has already, before coming here, had some experience on working on this kinds of models. So he joined IMSC 2012. Yeah, so it's been 12 years now. Yeah. So Shakti has been effectively, running our group for the past few years. Keep saying, I give the talks and Shakti actually runs the group. So very fortunate to have him, and I think we make a good working team. So his interest span as wide as mine, of course, and apart from pattern formation, he's been involved in a whole lot of projects, cell signaling and epidemiology and so on. So without further ado, let me invite Shakti to give the talk.
Thank you so much for that really wonderful introduction. So I hope for all of you are still got some energy, and I hope you're I hope you enjoyed the talk. I will be talking to you today about some work that I've been doing along with Chandrasekhar, who you'll be hearing from next week. He'll be giving a couple of talks, a couple of online talks. And of course, with work that we did jointly with Chitabra. The title of my talk is Embryonic Semaphores. Now, this is a little bit of a loose analogy, but I thought it's kind of fitting in with the theme of the workshop. Semaphores are basically signals that that can be you interpret to sort of derive some meaning from, right? And in some sense, that's what's going on inside the embryo. Cells are receiving all sorts of signals. There's a complex process through which they interpret this information in order to make the right decision. As you've heard during the course of this week with so many fantastic lectures. There's a really, really sort of fascinating and complex process through which this happens, right? And some of one of the kind of key ingredients in this is, of course, morphogens. And morphogens, we've already had a couple of talks on how morphological gradients are formed and so on, and how they've been interpreted. And I'll talk a little bit about that. This plays a kind of a key role in some of the work that we're doing. But another aspect which perhaps has not been touched upon too much up to this point is contact-mediated signaling. So typically in these sorts of early stages of development, the cells are fairly sort of close to each other, right? And they're kind of tightly packed, and so communication between cells is something which could potentially play like an important role in terms of its deciding upon its eventual fate, and that's something which we're going to be exploring in this talk. Sorry. Yeah. So we've already heard a fair bit about position information, right? So just to recap, when you have a developing embryo, cells basically need to decide what their eventual fate is going to be, right? A cell needs to know how exactly the course of differentiation should proceed. This needs to happen in a very sort of precise and robust manner, right? So you can't have cells which should be part of the limb for sort of taking on a fate which is of another part of the body, right? The cells need to sort of arrive at fates which are consistent with their surroundings, right? So cells that going to form that going to eventually form a certain part of the body, they should all be together, right? And so in other words, cells need to know where they are in order to know how they're going to differentiate, right? But of course, cells are, I mean, they only have access to a limited amount of information, right? So how exactly do cells know where they are, right? That's really the key question over here. So only if they know where they are, can they know exactly how they should proceed in their differentiation. And so this is what the concept of position information, this is where the concept comes from. So is that cells in these developing embryos receive some external cues, I mean, which is obvious. The cells are constantly sort of receiving signals from its environment, but these external cues are at least some types of external cues help them to sort of identify their positions within some coordinate system, right, relative to some or with respect to some boundaries, and then interpret that information to achieve distinct fates, right? And we've heard about morphogen gradients, right? So morphogens basically are some types of signaling molecules, right, which are produced I mean, they can have different types of morphogens, but nevertheless, morphogens are produced, in certain regions. They, as we've heard, about this production, diffusion, degradation model. So they produce, they diffuse, they degrade, and they form these gradients, right? So these morphogens are what essentially help provide the cell information about where they are relative to the source, right? So depending on the concentration of morphogen that the cell encounters, they can make an estimation about how far away they are from some boundary, right? And so this is like very roughly speaking, this is the key idea behind position information. So I mean, here's just an example from the Xenopus embryo, right? So you have activin, which is sort of produced towards the dorsal end, and sort of you have a kind of a rough gradient going from dorsal to ventral. And those cells which are kind of closer towards the higher concentration of activin, they will likely sort of end up in a certain fate, whereas those towards the other end where the activin is much reduced is likely to form another fate, right? So is this process by which cells attain different fates depending on the amount of morphogen that they sense is really the key process, right? Now, as I mentioned in this talk, this is a process that is subject to a lot of variability. So this is an inherently stochastic process, right? I mean, the morphogen gradient is sort of diffusing. There may be sort of its production itself there may be some stochasticity associated with it. The interpretation of the morphogen is also subject to variability, the various intercellular processes and so on. So there's an abundance of stochasticity in such systems, but nevertheless we find again and again organisms always manage to form fairly clear, precise, robust boundaries between different fates, right? How does this happen? And this is one of the questions which we're going to be exploring a bit later in this talk, but not right away. Okay, so here are just some examples of what morphogens can help to achieve in developing embryos. So, for example, here's the dorsal protein, which is sort of produced or which is found in abundance towards like this one side, and then like towards the ventral side, and then you see that it's sort of there's a gradient towards from ventral to dorsal. But concurrently, you see that there's sort of higher expression levels of twist near where dorsal is high, and rhomboid where dorsal is low, right? So the morphogen, the expression levels of morphogen are playing a role in some downstream process, which is giving rise to variable expression levels of some further genes downstream, which will have some subsequent role in patterning, right? And similarly, like in the case of the bicoid protein, which going from anterior to posterior, you see that you have one gene here, orthodendrical, expressed at higher and hunchback at lower levels of this protein. So, I mean, you can see straight away that, I mean, this plays an important role in establishing polarity and also in patterning, right? So, so morphogen gradients are extremely important for these very sort of fundamental sort of fundamental processes in early patterning. So. A question that one can ask is how exactly does one kind of tune a flag, right? Okay, so maybe I should have mentioned I mentioned the word flag, but without talking about the flag. Maybe let me sort of describe that briefly. So we've heard a little bit about the French flag model, right? From Wolpert, that was proposed by Wolpert. Now, the idea, I mean, just to reiterate, is very simple. If you have like some concentration of external concentration of morphogen, then cells, depending on the threshold of the level of morphogen, they can make some decision as to whether or not they will achieve a certain fate, right? So, for example, here we see that there is some profile of the morphogen gradient concentration, and you see that in this case there are two thresholds that have been established, right? And so cells in this region are seeing morphogen that are above this threshold. And this is triggering something for the downstream, and they're achieving this fate over here. Cells in this region, the concentration is above this threshold, but below this, and so you reach this threshold. And finally, these cells, they are seeing a concentration which is even below this threshold, right? Now, so this is what was what he termed as the French flag model, right? I mean, so because this was just a very simple sort of scenario where you can have, like, just with two thresholds, you can have sort of a patterning of three different domains, right? But the question is how exactly does one tune this in such a way that you can achieve domains of required of desirable sort of sizes, right? I mean, essentially, like you would want to have some control over how exactly you set this thresholds, right? So this one and this one, depending on how you set them, you can get like different outcomes. So in this case, in this sort of schematic which is shown over here, these two thresholds are set in such a way that these domains are roughly of the same size. But here you can see that you have a lot of variability in the domain size, right? So you can see here the is mostly dominated by blue, by white, and so on. How exactly do you sort of tune that, right? But of course, one way to do that is by sort of modulating the threshold itself, right? But that comes with its own issues. I mean, how exactly do you robustly, or do you sort of accurately sort of tune the threshold itself, right? You have to have some process by which sort of dynamically kind of can change the threshold to a desirable level, right? And another sort of slightly related question is that how do you sort of end up with these sorts of like pathological outcomes, right? So for example, here if you have sort of a gradient like this, like how do you sort of what are the situations that would give rise to some sort of like, so here you see that we have sort of lost one of the domains. But we are sort of having, like here we're having like stripes of red, blue, red, white, red, and so on, right? So putting all this together, like how do you, is it possible to sort of vary the size of these cells, these sulfate domains in a sort of a regulated manner, like in a way that, which we can just tune the domain size to in a way that is desirable for that particular process, and also while doing so, make sure we avoid having scenarios like this, right? So the idea that we had was perhaps cell-cell communication or intercellular communication could be playing some role in this. And so that's what we'll be talking about first. So how potentially could intercellular communication play a role in helping us to achieve these sorts of outcomes where we can tune the pattern dynamically, right? Now, and how do we do so in a way that makes sure we don't get pathological outcomes that don't preserve the sequence and the number of domains. So this is the basic question that we're asking. So it's well known that in these contexts, one predominant way by which cells communicate with each other is through the Notch signaling pathway, right? Essentially, how this works is you have cells which are in close proximity with each other, and on the surface of the cells you have receptors. Right? So these are these Notch receptors, which are these kind of hooks, drawn here as hooks, right? And you have ligands as well, right? So these could be like delta, jagged, and so on. But you have some ligands on the surface. When you have a successful binding between a ligand of one cell with the receptor of its neighbor, now what happens is that the sort of intracellular domain, which is the NICD, Notch intercellular domain, so that will just cleave off from this complex, and it will translocate to the nucleus, and then further activate some target genes, right? So this is the process by which it works, and what's not shown here is also is that the genes themselves can promote or suppress the expression of these receptors. So essentially, you have some way by which the gene regulatory activity of one cell can influence that of a neighbor, and vice versa, right? So that's how you get communication between the two cells. Right? And of course, if you have cells in a tissue and all of them are sort of close together, you have a lot of interactions, and so you can have so this can lead to some very interesting sort of outcomes, right? In terms of how they talk to each other and how they communicate and reach a desired kind of collective outcome. So I'm going to just leave this up for a second, not to scare you, but just to sort of give you a picture of where we're going. Right? So don't worry, I will break this down and explain all the components of the model. But this is how we sort of envisage this process, right? Essentially, what we were thinking was that, let's say you have a cell, right, and you have some circuit which corresponds to like genes, upregulating such down-regulating each other. In this case, we have taken 3 genes which sort of will correspond to like so when you have one gene which is dominantly expressed, it perhaps would lead to a certain fate. So which is why we have called them B, W, and R, which means that one of them would be like the blue fate, one of them the white fate, and the red fate, right? Now this particular model, this particular circuit, I'll explain in a second why, or I'll mention in a second why we've chosen this form, but so put that aside for a second. The idea is that you have, within each cell, you have some sort of gene regulatory network. You have receptors and ligands on the surface. They can bind to that of a neighbor. And then you have the feedback. So now the genes can promote or suppress the ligands, right, on the surface, and similarly, when you have successful binding of Notch to a ligand of a neighbor, that can in turn sort of affect the genes. I'll come to all of this. So this is just to give you the idea, right? And of course, we also, because we're looking at the effects over space, we're also looking at the morphogens, which are forming maybe some external concentration gradient on the outside, and then they are affecting the genes. I'll come to all that. But anyway, but the point is, so this is the sort of framework that we envisaged, and so I'll go through them now, like individually. So first of all, as I said, we have the 3 gene interpretation module. Now, the one that we have specifically considered is from this paper by Velasquez et al. Right? And so this is so in the presence of a sonic hedgehog morphogen, you have these three genes, PAX6, OLEG2, and NKX2.2. I mean the details are present here, but basically this is the way that they sort of repress each other. Okay. Now the blue or the PAX6, this is occurs even in the absence of the sonic hedgehog, right? So in our simulations, we will start with sort of an elevated level of B, and these two will be basically absent, and we'll see what happens over time, right? We also have, as I said, morphogens, right? So here the sonic hedgehog. So this will bind to some receptors on the surface, and then they will upregulate the here the W and R. Just for simplicity in future, I'll just call them B, W, and R, right? So the W and the R, but this is what they correspond to. So okay, so this is this is that part of the module, and so how it's modeled. So again, the model. So this is just the model as presented in this paper. We will modify this, but in the paper by Velasquez et al., this is how they had modeled it. So essentially, you have three equations for the expression levels of these three genes, B, W, and R. And so you have essentially like the production of B is going to be inhibited by R and W, right? So going back to the circuit, remember, it's inhibited by R and W. Yeah. So it's inhibited by R and W. And of course, you have in all three cases, you have some decay rates. So K1, K2, and K3 are the natural decay rates of your three genes. And W is inhibited by R. Go back and see W is inhibited by R, not by B. And are inhibited by both B and W. Yep, are inhibited by both B and W. So that's that part of the model. And and then also, this is the effect of the morphogen, right? The morphogen is going to is going to upregulate the production of W and R. So that's shown over here. And so basically, this is it's sort of saturates. So when S becomes very large, it just sort of saturates to beta, right? And to gamma. So in this model, they had just left this morphogen level as a free parameter. So SM is the morphogen level, and they just left that as a parameter. And I think they called it G in their paper, but there was like a free parameter. But since we are also looking at the effect of what happens over space, we thought we should model this. Using sort of an exponential function, right? So in our simulations, we considered like a linear array of cells. So we took the 30 cells, and its concentration profile is sort of decaying. So here going from right to left, you see that so for simplicity, we're just going to take a functional form like this. But in the second half of the talk, I'll mention a way by which you can actually dynamically sort of generate the production of morphogen. But in this case, it didn't matter all that much. So we're just going to assume like an exponentially decaying profile of morphogen concentration, right? So at cell 0, this is what the concentration is, and then it just decays over space, right? So fairly simple. So without accounting for any couplings so far. So this is just basically the original model by Velasquez et al. With the additional sort of spatial component to it. So now you have in this top plot, you have the morphogen concentration sort of decaying over space like this, going from, so here the axis is going from right to left, so going from 1 to 3, 4 up to 30, right? So we have that's a morphogen, and down below we have the so Y basically it just corresponds to the expression level of one of the three genes. So here it's B, W, and R, right? So you see that after some time, well, actually, let's look at the figures on top first. So on top, you see that in all three cases, we start off with some elevated levels of B, right? So remember, the B is is present even in the absence of which B is the PAX, right? So it's present even in the absence of sonic hedgehog, right? So this is high initially, and then you see over time the other two start to increase. When with the morphogen is very high, what begins to happen is that you see initially you have the white sort of rising a bit, and then the red dominates, and so that's what happens over here in this region. In this region, the white just dominates, and over here the red and the white they never reach a sufficiently high level, so the blue continues to dominate, right? So you see it's a very kind of clear sort of outcome, right? So blue, white, and red is dominating in like 3 different regions, and we've tuned the parameters in such a way that we have like a 10-10-10 sort of breakdown, right? So I mean this is just to give us a sort of a benchmark, right? So so this is what you'll see in the absence of any interaction. So now the question we ask is, all right, fine. So far so good, but now we want to see, is it possible to now tune the flag? So that's what we want to try. So for this, what we do is we say, all right, so let's just have notch ligands, so notch receptors and ligands on the surface, and wherever you have a bound notch complex, so you have basically notch binding to ligands of a neighbor, then it can have some effect. So basically the NICD, as I said, will cleave off, translocate to the nucleus, and it'll have some downstream effect on the 3 genes, right? Now, I here I've marked these as like sort of open squares. And the reason is because we're still keeping a sort of ambivalent as to whether or not it's upregulating or downregulating. It's doing something, right? In principle, like this could upregulate while these downregulate. All 3 of them could downregulate. All 3 of them could upregulate, and so on. So you have a lot of different combinations, right? So I've just kept it general for the time being. And this is how you would modify the model, right? So basically, what we're doing here is we have these additional terms in the numerator and the denominator, where we say that we introduce these parameters phi 1 2 3 and xi 1 2 3 times basically so NB is the concentration of so this is basically like NB is a proxy variable for the NICD, right? And L is the ligand concentration. So of course L is not coming into this equation as yet, but so these are the 2 new species that we've added. So in the numerators, you would have phi times NB by some threshold, right? So when it crosses the threshold, that's when it's activating. And similarly, we have downregulation as well in the denominator, right? So you notice here we have both upregulation and downregulation. Obviously, that is not. It can happen, but it's not exactly what we want. But we've written it this way because now it gives us the flexibility to use the same model to upregulate or downregulate one or more of the genes at the same time. So we wrote this in such a way such that we can do a lot of different things with the same model, right? That's why we've done it like this. And I'll come to all these various combinations later. So that's the first half. So now remember there's also the feedback. Right? So the expression levels of the genes is also going to have some impact on the expression of the ligands, right? So it could promote or suppress the ligands, right? And the ligands will in turn, it can bind to the notch of the neighboring cells, right? And so again, so these 3 squares could be like a positive or a negative kind of interaction. And this can be modeled once again in a fairly simple way, right? So we have, remember the ligand, it could be upregulated by any one or all of the 3 genes. So we have this in the numerator. And so we have this is also phi, but this is a different phi, right? So we're calling it phi. And so this is zeta in the denominator, which would correspond to repression. And again, we have both of this in the same model because it just gives us the flexibility to do whatever we want. So we don't have to have like hundreds of different models. All right, and then the trans. So I think I didn't mention this. So basically, when you have successful binding across cells, that's referred to as trans binding, right? So the trans binding of notch receptors to ligands of neighboring cells. So this is the ligands of neighboring cells, as we refer to that as L trans. And in our model is basically just the total, like the sum of all the receptors of the neighbors, right? So that's what is L trans. So if you have two neighbors, you basically just sum up the L. And so that leads to the release of the receptors intercellular. Sorry, the intercellular, the NICD to be released. And so remember, NB, which is we're kind of treating it as a sort of a proxy variable for the intercellular domain. So this is so with more L trans, you have more production of NB, and these two are just like the decay for those two terms. Okay, now just to start with, I will mention just 4 cases, right? So in principle, all 6 of these links can be either positive or negative or even nothing. Like, I mean, they could even just do nothing. So, you have a lot of possibilities. But just for simplicity, we're going to look at 4 cases. Right? So the first case is where the notch intercellular domain is going to have it's going to inhibit the activity of B, W, and R. It's going to inhibit all of them. And similarly, B, W, and R is going to inhibit or to repress the promotion of the ligand, right? So this is what we refer to as type 1. And in all the other types, we assume that basically the NB is going to have the same effect on all 3. So either it's going to downregulate as it does here and here, or it's going to upregulate as in here or here, right? And similarly, the 3 genes B, W, and R are going to either repress or promote the production of ligand, repress or promote. I'll come to a slide a bit later where we start to explore all the possibilities, because just to begin with, we want to keep it simple and just look at scenarios where the notch has similar roles in terms of how it affects the genes and how the genes affect the ligands. So, but this itself will give us some interesting insights as to what could be happening, and then we'll expand that to all the possible cases. Okay. So anyway, so to start with, like I said, we're going to have these 4 coupling types. Okay, so I guess you don't need to worry too much about it. This is more just an issue of notation, but going forward, we're only going to talk in terms of theta 1 to theta 6. But remember, in the model, we had all these parameters. We had all the phi 1, 2, 3, psi 1, 2, 3, and so on, phi 4, 5, 6, and zeta 4, 5, 6. What we've done is basically we've just, we say that, so whenever you have downregulation, basically, so that means that phi, this is another phi. Basically, the phi's are 0. So whether this has been downregulated, this has been downregulated. But if you have upregulation, we're having zeta is equal to 1, or sorry, psi is equal to 1 or zeta equal to 1, whereas the phi's are more than 1. Essentially, what we're going to do is we're going to just write this in terms of a set of four values, but that will correspond to the phi I, psi I, and the other phi j and the zeta j. But like I said, maybe let's not worry too much about this. The point is that these 3 thetas are going to relate to, I think, maybe this is the simple way to think about it. These 3 thetas, 1, 2, 3, suppose you're having, suppose your interaction is negative, then, or it is negative or is inhibitory, then what you're going to do is you're going to set this phi to 0, and you're going to set these psi's to some values of theta 1, theta 2, theta 3. And suppose your interaction is positive, you do the opposite. Basically, you set these to be theta and this to be, well, here you're not setting it to 0, setting it to 1, right? I mean, if it was purely symmetric, you set this to 0, setting it to 1, because we wanted to have a sort of like a kind of saturation, right? So anyway, so but let's put that aside for a second. But the fact is, one can set these one can describe the entire parameter space strictly in terms of the thetas. Now, what the thetas correspond to is sort of context dependent, and this will actually make a little bit of sense later when we have the like an entire sort of set of all possible interactions. So for the moment, just note, all you need to know is that we have 6 parameters. They could be positive, they could be negative, but that's very much dependent on the context. Whether it's here, for now, we have 4 types, and you have the parameters set accordingly. Okay, so let's move on from that. Okay, so now let's just explore what's going to happen in these 4 cases, right? So here are just some sample sort of simulations for what you would get if you have these 4 coupling types. So what do we see is that in the first 2 cases, so this is type 1 and type 2, right? So what we call type 1 and type 2, and they are characterized by the notch intracellular domain having a suppressive role on the 3 genes, right? So in both these 2 cases, the genes are suppressed by NB, the variable NB. So what we see here is the way to read this figure is that, so this is space, right? So along this axis, that's space. So we're having our 30 cells, and along this axis, that's time, right? And along the vertical axis, that's the level of one of the 3 genes, B, W, or R. Right? And what we see is that, and remember, we're always starting with higher levels of B, right? Because B is present even in the absence of any morphogen. So regardless of where you are in space, B is sort of initially much higher, right? So you start with B high, and then over time you see that B starts to sort of like get kind of it starts to reduce, close to the source of the morphogen. So remember, the morphogen is going from 1 to 30, right? Close to the source, you're seeing that eventually you see that the red, I call it the red gene is going to finally have a increased expression level. Then over this region, you have like the white gene is dominating, and then finally over here, from let's say around like 17 or 18 onwards, the blue gene is dominating, right? And actually, what we're showing on top over here is just basically, it's the same data, but we're just showing it as sort of like a kind of a space-time kind of plot. So basically, you see that at each, so here it's just initially it's like, okay, so the color basically means which gene is dominating. So if you just see white, it means white is dominating. Blue means blue is dominating, right? So here, like blue is just always dominating from here. So white or red never takes over. Here, white's always dominating. Here, white's dominating for some time, and then red takes over. So that's how you read this figure on top. Okay. So right. So type 1 is quite straightforward. What we're seeing, of course, is that you see that now we're having a slightly sort of expanded kind of region of white. Right? So the red is sort of reduced a little bit. But this is just one sample, one example. So, but this is just to give you an idea. And similarly, something very similar over here as well. Right? So basically, you have like red where the morphogen is high, then white and blue. What we see down here is a little bit different. So here, remember, so what is common between these two is that notch is upregulating B, W, and R, right? The difference is what the genes do to L. So here they repress it, here they upregulate it. But here you see that you have a slightly different outcome. So over here, of course, the blue dominates, but initially you see white is dominating for some time, then blue dominates. And here white dominates. Here you see red, and then over here once again white is dominating, right? So you have basically we now have four domains rather than 3, and over here it's even worse. So here you're having like white, red, and so on. So it's complete kind of a mess in what's going on over here, right? So okay, fine. So this gives a rough idea of what the different coupling types are doing. And here just some more examples, right? So here remember type 1. Now here I'm just showing 4 examples in each case. You see that, all right, so you have the same sort of chromatic order, right? Basically the blue, white, and red, but the widths of the regions might be changed slightly, which is kind of what we were looking for actually. that basically you can just tune the widths of these regions. And the same is happening in type 2 as well. But in type 3 and type 4, well, here it's okay. Here actually you're having three regions, which is kind of okay, what you'd expect, but not here. Here you see the blue is gone. And over here now you have a white, blue, red, and white. And here once again, it's complete chaos. So over here, it's like you have a very large number of like alternating like red and white regions. So so this is just giving you a rough idea about what the different coupling types are doing. But what about like if you take a you have a question.
Cases, what's the difference between the 4 plots? Like what's different between the 4 plots? Next slide.
Oh, between these?
Yeah.
These are just different. Yes, I didn't mention this. I'm so sorry. The values of theta.
Okay.
The values of theta, I mean, we've taken them from like a distribution, and actually this becomes very clear in this plot over here.
Right.
So here, what we've done is, so remember the we have some freedom to play around with the values of theta, and we can choose them to be different, right? So suppose we take so here we've taken 10 to the power 5 parameters, in each case 10 to the power 5 parameter sets where of all the thetas, where each theta k is sampled from this range 1 to 10 for the activation parameters. So whenever you have something like this, right, so like activation, then it's from 1 to 10. And whenever it's something like this, so it's sort of inhibition, then you take it from 0.1 to 1, right? So whether it's activation or inhibition. And what we are showing is basically along the two axes, we are sort of displaying the result. Okay, let me just say what's along the z-axis first, then I explain the other two axes. So along the z-axis basically is how many times are you seeing a particular outcome, right? That's what a particular outcome, and the outcome in particular is basically measured in terms of these two measures basically. So the DH and NB. The NB is just strictly the number of boundaries, right? So here you see we have, so NB is 2, right? 2 boundaries, which is what we're seeing in all these cases, right? So in all these cases you have blue, white, red, 2 boundaries, right? And out of all the 10 to the power 5 trials, you're basically always seeing 2 boundaries. Okay, so that's what happens in type 1. In type 2, most of the time you're seeing 2 boundaries, so NB2, but sometimes you're seeing also 4 over here. You see that it's happening just a small amount of times, but for the most part it's 2 boundaries, right? And what we're just showing over here on the side are just some samples, just some randomly chosen samples of what the flags would look like, right? In terms of the how the relative sort of lengths of the relative sizes of the different domains. What we're seeing here, right? So we already knew that before coming into this that types 3 and 4 are going to give us some very odd results, right? So how do we quantify that over the entire ensemble? Well, let's just first look at the NB, the number of boundaries. We see that here we have sometimes one boundary. One boundary, it just means that you just have 2 domains, right? That's what, and so we're getting that quite often, and so we're also getting some at 0. Right? 0 just means you just have something like that. You just have one color, right? So 0, 1, 2, sometimes 3 as well, and rarely 4. So here we have 3, 1, 2, 3, and say 4, right? So you do get that. It's not as often, but you do get that as well. Oh, I'm sorry. I also forgot to mention DH. So what is DH? Right. So DH. DH is basically the Hamming distance of the obtained flag to the idealized flag that you would get in the absence of coupling. So we already know what happens if there's no coupling, right? So going back a few slides, we know that we've tuned the parameters in such a way that for our choice of parameters, we're going to get a scenario where you have like blue of length 10, white of length 10, and red of length 10, right? So we're going to get this outcome. So if we have that, you can actually, so if you treat that as a string, right? So basically blue, white, red, you treat that as a string, and you treat these as strings as well, you can just look at the Hamming distance between the strings. So for those of you who are not sure what having distance is, so suppose you have, let's say, AAA and AAB, right? So these are two strings, so the same length, right? The only difference between them is that, so in one case, I'm ending with A, one case I'm ending with B. So that has a Hamming distance of 1, right? So if you suppose you have AAA and BBB, you have a Hamming distance of 3, and so on, right? And so in that sense, you can look at the Hamming distance between the entire string of 30 sites, so which is why it can go up to 30, right? So the maximum Hamming distance is 30, right? So you see here that it's sort of spread a little bit, right? So basically, and so the peak is somewhere like around 8 or something like that, right? But it's not a lot. I mean, you can have some variation from the original flag, but some variation is also good, because we also do want to be able to tune the flag a bit, right? So so having a larger data is not necessarily a bad thing, right? So that's what we're seeing over here, and so here so here too it's fairly sort of narrow. Here you have a huge, I mean, as expected, you'd have a huge sort of range of DH. I mean, the Hamming distances, and look, and type 4, for example, the one where we were getting all sorts of boundaries. I mean, unsurprisingly, this is quite a mess. I mean, you're having up to 9 or 10 sort of boundaries between the colors. You're having a whole wide range of Hamming distances and so on. So yeah, this is as expected. So yeah, so that's what that gives us sort of a macroscopic sort of overview of the results. Now, before I get to what happens when you sort of open it up. So right now we looked at just 4 coupling types. So we're going to open it up later. But before I do that, I'll talk a little bit about little bit of analysis that we did on the sensitivity of the results to changes in these parameters, right? And so for that, what we did was we used this Sobol method, which is basically a variance space sensitivity analysis that we used to quantify the contribution of the parameters to determining the cell case. And so what we did was we considered the final state of each cell to be represented by some F, which could be just 0, 1, and 2. Let's just, for simplicity, I mean, so it would be basically B, W, and R, but we'll just call it 0, 1, and 2 just for simplicity. And at each site I, we just compute we first compute the variance in the F of I across realizations, right? So basically, let's say you say site number 1, you're going to just look at what is going to be the fate across a whole bunch of realizations. So for example, if you're consistently finding it's blue, 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 the variance is going to be 0, right? But suppose you're going to be sometimes blue, sometimes red, sometimes white, you're going to have a higher sort of variance, right? So that's the first thing we do. The second thing is that we measure this, what's called the first order sensitivity index, S1, at each site I. And we do this by computing the variance of this measure. So I'll just take a minute to explain this. So basically what we do here is to say that, let's just say we have 6 parameters, theta 1 to theta 6. Let's just say I'm having one of the parameters. Let's call it, let's say, theta 3. Okay, so let's say J is 3. I'm going to fix my theta 3. And I'm going to see, okay, for this theta 3, what's going to be my value of F? Basically, I'm going to get a blue or a white or red on this side. Now, that is also going to depend on what my other parameters are, theta 1, 2, 4, 5, and 6, right? So I have choices as to what I'm going to, I have different choices for those parameters. So what I'll do is basically take an ensemble of all those other parameters, fixing theta 3, vary all those other parameters, and then in every case, find my F of I, right? Just take that average, and then take the variance of this across all the thetas, basically. I mean, of all the possible values of theta j, because so theta j, of course, here I'm saying theta 3. So now theta 3, remember, I can also vary that as well. So I'm now taking the variance across different values of theta 3. So first I'm fixing theta 3, taking an ensemble of all the other values and getting the average. then I'm taking another value of theta 3, doing it again, another value, doing it again, and then taking the variance across this. So okay, hopefully that made sense. And so that's this first order sensitivity index, and that's normalized by sigma square. So anyway, so this is what we get. So first of all, the variance itself. So perhaps unsurprisingly, for types 1 and 2, you see that it's almost non-existent everywhere except near the boundaries, right? So far away from the boundaries, you see that there's almost no variance. So if you're going to get blue once, you're almost always going to get blue, and similarly for white and so on. Well, white is a little tricky because white's over here. So white, you're going to be, whether you get white or blue, it's there is a bit of fluctuation. But in types 3 and 4, you have variance all over the domain. So anything, it's up for grabs. Basically, what color you get is up for grabs. Now, what's shown below is the S1, right? So the S1 is the measure that I had talked about earlier. And so remember, this measure you can do separately for each parameter. Basically, how much is that parameter contributing to the variance, right? And so what we were seeing is that really the parameters that contribute to the variance are, and so we've colored them based on what they are. So theta 1 is green, pink, and so on. You see that by and large it's theta 2 and theta 3, right? Again, here it's theta 2 and theta 3. The others have a very sort of small influence, but it's these two parameters that are playing the key role in terms of accounting for where is this variance coming from, right? And in the other cases, it's, in these two cases, but perhaps not so interesting because we already know that it's sort of, there's so much variability. Interestingly, theta 1 and 2 are still playing a big role, but now theta 1 is also playing a role. So theta 1, if you recall, is basically what notch does to blue, right? The notch bound does to blue. So theta 1, 2, and 3 are playing the key roles over here. But yeah, so let's just explore this a bit further. So remember, theta 2 and theta 3 are playing a really important role. Surprisingly, 4, 5, 6 are doing almost nothing. So let's look at this in more detail. So in type 1 and 2, where some interesting stuff was happening, what we can do now is we can similarly, so just like how we obtain the S1 by looking at how the color was changing, we can also use the same analysis to see how the length of the domain is changing. So for a given set of parameters, we can see how this is affecting the lengths of the domains, right? And without getting too much into it, basically what's shown here in these pie charts is basically how much this index is described in terms of each parameter, right? So you can see that, and the three rows, the outer one is basically the length of the red region. So LR is the red region. The middle one is the white region, and the outer one is the outer is red, then the middle is white, and the blue is the inner one. But regardless, the point is that it's dominated once again by theta 2 and theta 3. So theta 2 and theta 3 are really playing a key role over here in both these two cases. And so these two figures are just to say that how much what is the likelihood that you're going to see, or the probability, the joint probability of seeing a given value of, or the given sort of length of the white region and the red region. You see that it's in both cases it's sort of distributed pretty closely around like 10-10. So 10-10 is where you have the perfect or the ideal flag, and you have a little bit of variation around that, but not too little and not too much.
I'm really sorry for interrupting.
Yeah.
So I just want to understand what you exactly mean by the contributions of theta 2 and theta 3 being more. So in the previous graph, the pink and blue bars being high. So is that a reference to the quantity you mentioned earlier, the F theta J quantity, or is it something different? So I don't remember the name. So on the previous slide, so first order sensitivity.
Yes.
So is it that which is being plotted over there, or is it something?
And I kind of I went over it a little bit quickly because there's a lot of information, but I mean I would explain, but it's a good question. So this is this index. You obtain this separately for each of the parameters that you're varying, right? Now, in what I didn't mention, that in general, what you can measure using this approach is the effect of varying one parameter at a time, two parameters at a time, three at a time, and so on, right? And in principle, all of it together will tell you like the total variance, or the effect of the total variance. So it should all sort of sum up to to one. But here we're only looking at the variance one parameter at a time, right? And so what we're showing in this figure is, and so you see it's not summing up to one because I'm only looking at one parameter at a time, right? So what it's telling you is at this site. So basically, maybe again I should have explained this, is that we are calculating this separately at each site, right? And at each site you can say, okay, so over my ensemble, what are the different colors? The F is basically, you can think of as the colors. Basically, I'm calling it 0, 1, and 2, but these are the three colors. So what is the variability in those colors across that site, and how much of it is how much of this variability? So the variability itself is given by this sigma square, right? That's itself is the variability. Yeah. But how much of that is coming as a consequence of changes in each parameter? How much of it is coming because of changes in theta 3? So if you notice, let's look at this, for example. You notice there's almost no contribution of any of these colors, which means that essentially, suppose I had fixed these three parameters and I had just changed these three, I'd get basically no change.
Right.
I'd get basically no change. So, but suppose I had fixed everything except theta 3, right? And now I'm changing theta 3, I might start to see some changes. So that's kind of what it's telling you. So what are the important parameters in terms of looking at the contribution to the variance in the outcome? And here the outcome we're specifically focusing on is F, which is the color. But you can look at something else also. Yeah. So so hopefully that answers. But thanks for the question. I mean, I probably should have mentioned that in detail. Okay, so I think I'll move on from this because this is getting a little bit into the weeds. But just to mention one more thing, is that here what we can also see, because remember theta 2 and theta 3 are the two parameters that will seem to be like the most important in this case, right? So variations in theta 2 is seeming to have like play a big role in terms of the field that you're going to see over this entire part of the domain, and theta 3 as well, right? So what is that doing to the actual location of the boundary? So what we're plotting over here is the location of the red-blue boundary, so the red-white boundary. So this surface is basically the boundary between red and white. So you can imagine basically below this is all red, right? Now between this surface and this surface, it's all white, and above the blue surface, it's all blue. So you can just think of it. I mean, that's like it's a neat way to think about it. So what we are seeing is, first of all, as you and so in type 2 and type 3, as you increase the parameter theta 2, what that does is that you see that the red starts to increase a little bit, and the blue starts to decrease a little bit. So what that means is that the white region gets sort of squashed a bit, right? So both the red region increases, the blue region increases, and the white one gets a bit squashed, right? And that happens both here and here as well, although not so much over here, but it happens. Increasing theta 3, so basically going from here to here, you see that makes a big difference in terms of the R, right? So it expands the sorry, actually should not expand it. So I think it should be shrinks. So yeah, I'm sorry. This should be it shrinks the R region with almost no change. The B region, you see, is completely flat over here. As you change theta 3 is completely flat, right? Yeah. So so this is sort of like at a sort of a final level detail telling you what exactly theta 2 and theta 3 are doing to the patterns, right? So this at least we can sort of extract this insight from these results. Okay, now as promised, I will now open it up. So till now we've been looking at just 4 coupling tests, right? So, but in general, you can have each of these 6 links positive, negative, or doing nothing, right? So you can in fact have, like, say 6 links, so 3 to the power 6, positive, negative, nothing, 729 different classes. So obviously we didn't want to write down 729 different models, so we want to just have one model that can do all of this, and so that's why our model looked a bit complicated, but it's for a reason, right? But yeah, so in principle we can study all of these classes, right? So so here are just some examples. So for example, in this case, we're having theta 1 is negative, 2 is doing nothing, 3, 4, 5 are negative, and 6 is positive, right? So just one example. You can see in this case, it's kind of similar to what we're seeing in, if you remember, type 1 and 2 look a little bit like this. And here's another, I mean, I won't go into detail, but here's another example where here a lot of them are positive. And, you see this is a little bit similar to what we're seeing. I think it was in type 3, right? So this is just to give you some idea about, this is, for each of these cases, you can study the outcomes. But of course, to get 729 images like this is going to be, I mean, how much are you going to actually be able to extract from that? I mean, it's just, we need to be able to look at that in a little bit more of a succinct kind of manner, right? So that's what we're looking at over here. So here we're saying, okay, fine. So each of these parameters, 1, 2, and 3, 4, 5, and 6 can be positive, negative, or, basically it's not doing anything, right? And each of these parameters we can take from a distribution, do an ensemble, basically take an ensemble, do some simulations in each case. So this is actually obtained from like a very, very large sort of set of simulations, because for each choice, we're doing a large number of simulations, and we are looking at for over the ensemble, what is the fraction of times that we're getting a French flag. A French flag is, how we're defining it, is basically a flag that has 3 distinct regions occurring in the expected chromatic order. So we're just keeping it very, very sort of achievable. We just want that, and it turns out that very interestingly, it looks like it has almost no dependence on what the values of theta 4, 5, and 6 are. Right? So theta 4, 5, I mean, not just the values, what they're doing, whether or not it's a positive or a negative, it almost doesn't matter. You see that for all these rows, right? Like you see that you get like almost yellow, which means that you're almost always getting French flag or something very high, regardless of what is the particular sort of role of 4, 5, and 6, whether it's positive or negative. But it's very strongly dependent on the theta 1, 2, and 3, right? So when, for example, theta 1 is positive, you're almost never getting a French flag, right? Of the expected type. But in these cases, you are, right? So this kind of offers some insight as to where we should be looking to get the flags that we want. So okay, so I think I will stop with that. I probably went way over how much I expected to spend on this part, but at least hopefully that kind of gave you a kind of a flavor as to the kind of stuff that we were exploring over here. To put this part in a nutshell, basically what our results seem to be suggesting is that juxtacrane signaling or contact-mediated signaling seem to be playing a key role, or it could play a key role in adaptively regulating the cellular differentiation in these cases, and you can get the flag, the desired flag in a way that could potentially compensate for things such as mutations or things that affect the interpretation of the morphogen and so on. So this could be a way of sort of conferring robustness. So that's one interpretation that we had. Another sort of point that sort of we thought is interesting is that it kind of it sort of in a way integrates 2 different paradigms, like the sort of the boundary organized mechanism for pattern formation, which sort of involves like a pre-pattern being the morphosync gradient, as well as a sort of a self-organized mechanism which is coming about through interactions between competing species. Right? So anyway, I'll probably stop with that, and I mean stop this part with that. I'll have a bit more to say, but just before I move on, I just want to mention that we have two papers on this. The first one has a bulk of the results. The second one is more of an opinion piece, but it has some of these figures as well. And the second one, in fact, is where we kind of took the title of this workshop from. It's in the title of the paper. And if anyone is interested in the to play around with it themselves, there's the code on GitHub. You can talk to me later. I'll give you the link. All right. So I will probably breeze through the second part. Is there any questions at this point? I mean, maybe. Oh yeah.
In the models you made, you varied the parameters of, activation or repression, right? And you told how robust it is when it comes to these parameters. What about the parameters affecting the gradient itself, morphogen gradient? So how robust is the system when it comes to that?
Okay, so we, for the most part, did not vary the gradient. Okay, first of all, going all the way back. Right. So essentially that's the morphogen gradient, right? So we have a very, very simple form of the morphogen gradient. We just say that it has some high level over here, exponentially decays all the way down here, right? And then lambda m is sort of controlling now this can of course be varied, and if you vary that, then that's going to change the size of the different domains. But all that's going to do really, I mean, so we have varied that, but basically all it's going to do is basically just give you a different benchmark. So we chose it in such a way such that this was our benchmark.
Okay.
Right? So, and the point was, starting with this, in the absence of any interactions, what would interactions do? So, so of course one can change this, but then one just has a different benchmark to compare the results again.
Okay. The question just was, if I vary lambda, and then is it just the domain sizes are affected, or would I see a drastic change in the pattern itself? So in type 3 and 4, for example, you changed the thetas, and then you saw a drastic change in, say, the amount of domains, for example. So would I expect something like that if I just change lambda?
Not so much for type 1 and type 2. I mean, unless you really sort of, where is type 1 and type? Yeah, not so much for type 1 and type 2, unless you really change lambda such that you basically almost push one pattern to the one domain to the corner or something like that. So for the most part, it should have no real effect on at least the qualitative nature of what type 1 and type 2 are doing. So I think even type 3 and type 4, sure, it will have some effect, but again, it depends on what you're quantifying, right? I mean, if you're quantifying in terms of something like this, potentially there may be some change, but it might just be sort of shifted towards one side or something. I mean, one has to look at that. What happens if you really crank up your lambda? How is that going to affect something like this?
Yeah.
But I mean, it's not. I mean, for the most part, we've not been focusing on that. We've just been looking at just keeping that as a benchmark to compare everything else against. So,
yeah.
But I will come now to the other question of what would happen if you sort of change this dynamically. So maybe So with this, I'll just quickly kind of breeze through the second part of the doc. So I've been promising to talk about noise, right? So here. It's just an example of what happens in an early embryo, right? So this is in the Xenopus embryo, and so what you start to observe is that there's, over time, you begin to see like a boundary that begins to emerge between cells that express two different gene types, so two different genes. So here, goose-squid and brachyury. So you see that, so it's kind of shown quite nicely in this paper, that you see there's a very sort of nice clean boundary that arises between those that are expressing this and those that are expressing that, right? And the important thing is that this is quite robust. Like, you take a large number of embryos, you're always going to find that this happens again and again, almost at the same location and so on, right? And you don't have, and it happens in a very timely fashion. It happens, at the same time, post fertilization in all embryos. And so the question is what exactly is causing it to be so precise and so robust, right? So there must be some way by which the cell is managing to perform some sort of corrections, right? So that's essentially what we're focusing on in this second work. So if the cells were only looking at the morphogen concentration, Now, what would happen is, like, suppose you had a very noisy. So here, okay. I have actually taken these results from a simulation of the model, which I haven't shown the model yet, but this is just for illustrative purposes. So what we're showing here is that suppose we have a noisy morphogen concentration going from, let's say, 0 to 50, right? So you have a noisy morphogen concentration, and you have a system with like two genes, right? Gene A and B. B is expressed at high morphogen, A expressed at low morphogen. What you would find is fine. So close to the morphogen source, you have B is dominating. Far away, A is dominating, but in the middle, there could be some ambiguity. Right? So the different dots correspond to from our model different simulations. So we do a large number of simulations. We find that so in this region, so sometimes you're having B dominating, sometimes A, and so it's not super clear even where the boundary is. Sometimes the boundary is here, sometimes the boundary is here, and so on, right? So rather than have a kind of an idealized situation like this, you could be getting something like this. So sometimes the boundary is here, sometimes the boundary is here, and so on, right? But we know that this is not what happens, in most cases. In most cases, you get this, right? So how does the embryo sort of kind of ensure that you're having this sort of robust outcome time and again? That's what we're looking at. So I've already explained aspects. So I think I can probably go a bit quicker because I've already explained aspects of this in the previous model, so I can probably go through it a bit quicker now. But essentially what we have here is a slightly different framework, but with the same sort of components, right? I mean, the same players, you could say. In this case, we're looking at a simple scenario where you just have two genes, one expressed at high morphogen, one expressed at low morphogen, and they're sort of like mutually repressing each other because this is sort of a competition in this case of which gene is going to dominate. I'll get into the once again. This is just an overview. I'll get into the precise details a bit later, but this is just to give the picture of what are the what is in our model. We have the genes. The genes are upregulated by the morphogen differentially. I'll come to that as well. You have communication via notch signaling. Successful notch binding can lead to upregulation or downregulation. Basically, some regulation of the genes. And similarly, the genes can upregulate the production of notch, right? So I come to all those points, but this is just to give you the big picture. Now, the way that we model it in this case, because we have to account for noise, stochasticity, is that we have a set of stochastic differential equations. So we write down the deterministic components in each case, and we also have a noise term over here as well. And just for simplicity, we're just assuming that we just have so in each variable you just have some noise strength times the corresponding concentration for each species, right? So if species X is high, then you're going to have a high fluctuation. If it's low, in lower, and so on. So that's the way that we incorporate the noise. And so when we do simulations once again over like an array of cells, here we're taking 50 cells, so now what are the components of the model? So to begin with, here the morphogen is we're sort of letting it sort of evolve dynamically. So we say, all right, so we have the production of morphogen at some source, diffusion and degradation, and then this should lead to something like this. But I mean, it will be much more noisy. This is just a schematic sort of representation. You will have a kind of a noisy kind of version of this. And for that so we write it down as this. So basically, this f of m production, right? So basically is only being produced at site 1. So that's a delta function, only produces at site 1. Diffusion, and that's the degradation, right? Then when you have successful binding, right? So we say that there's some downstream effector. So in the previous model we had NB. Here we're calling it S, but it's the same thing really. So we have the downstream effector S, and so this is produced as a result of transactivation. So when you have the receptors binding, so the receptors R, when it binds to ligands of neighboring sites, you have basically that's when this gets released, right? And then. And all the terms in our model, they have some degradation. So I won't explain that further, but basically all these have some natural lifetimes, right? Okay, So this is the free ligands. So the free ligands, they are produced at some constant rate, and the production is suppressed when the downstream effector is sort of increased beyond some threshold, right? So when you have a large number of downstream effectors are being released, then that sort of acts as a kind of a negative feedback in terms of suppressing the further production of ligands, because it's almost like if you have a lot of feedback, so a lot of communication, then it sort of limits further communication. So it kind of acts as a kind of bottleneck, you could say, on further communication. Right. Now, this is probably like one of the most important assumptions in our model, which is that so remember, we said that the free receptors are going to be upregulated by the genes themselves, right? But we're saying that this is only done when essentially A and B, here are genes A and B, are expressed at sufficiently high levels, right? So here, the level, we're just calling it some threshold J. So J is a parameter in our model, right? So we're saying that when A and B are more than J, then it leads to the upregulation of these free receptors. And so we're kind of modeling that as a product. So both A and B should be high, right? And the reason is because if only one of them is high, then there's no tie break. I mean, basically, it's very clear who the winner is, right? So the notch only kicks in when there is some sort of doubt as to what should be the fate, right? Because it makes sense that the notch should only work in that case. Notch should not, for example, notch should not really be doing much here where it's very, very clear B is dominating, or here where it's very, very clear that A is dominating. Only here where it's not so clear who is dominating. Okay, right. And then finally, over here, we assume that the morphogen is upgrading A and B at different levels. So different thresholds. So we say that it upregulates A at some threshold K1 and B at some threshold K2, and we're saying that K2 is more than K1, right? So but what that should also mean is that very naively speaking, is that K2 is more than K1, so the morphogen should upregulate A as well. Right? So why isn't A dominating over here? To account for that, what we're doing is we are having B inhibit A more strongly than the reverse. So that's what ensures that B takes over here, right? So this would be what would get in the situation where there's no noise, right? So that is how we were ensuring that this works. Okay, good. So yeah, and finally, this downstream effector can upregulate or downregulate A or B. And what we're going to do is we're going to look at, for now, we're not going to say what it does. We're going to say, well, it can do one or the other. Let's just see what it does. So, right. So I won't go into the much details of the model, but just to say that once again, it looks kind of horrible because of the fact that we want to model 4 different coupling types within the same model. We want to account for all 4 of it. Essentially, if you want to have S inhibit A, S upregulate A, S inhibit B, or S downregulate or upregulate B, all you have to do is you just have to change these parameters accordingly, and then that would describe the scenario, right? Basically, so yeah, I mean, I don't want to get into step by step what happens, but basically you can sort of, if you just take a glance at it, you can see that that's what's going on, right? So all 4 of these types of scenarios can be accounted for by this one model, right? So okay, so now let's just see what happens. So in the case where let's just take this case where S inhibits B, okay? In the absence of any interaction, this is what you'd observe, right? So you have a lot of fluctuation, like near and far from the boundary, but and close to the boundary, you see that sometimes one gene is dominating, then another one, and then one, and so on. So it's not even really coming to a clear decision. But if you look at this case, when S inhibits B, first of all, you see that there's a reduction in the fluctuation, and secondly, there's a very clear sort of winner. So if you see here for a short while, you see this gene is dominating, then suddenly the other one takes over, right? So there's a clear separation between fate domains, and the cells converge very rapidly to their fates, right? And this can be seen even more clearly if you look at like the distribution over an ensemble. If you take a large number of simulations, you look at the distribution, you see that, well, in the uncoupled case, the distributions are kind of overlapping, which means that, it's not super clear like what the final level is going to be, sometimes high, sometimes low. But when, S inhibits B, it's clear separated. It's either A or B, right? So that's what's happening up here. So this is the high level. It's either A or B, right? And if you look at this picture, which is, if you remember several slides back, I showed you this picture. You see like it's very fuzzy over here, right? The same picture now looks like this. See? So it's become very sharp, and there's almost no ambiguity at the boundary. So the boundary, the location of the boundary has become much more like sort of localized. I mean, much more precise as to where the boundary is occurring. Right? Yeah, and what we're showing over here is is that these two parameters Q and J are related to the thresholds. So remember here we said that A and B should be above some threshold. We're just calling that J, and where is the other one? Oh yeah, sorry, here. So basically, and this threshold Q is basically is what and comes here as well. Basically, is what the threshold of the downstream effector on the regulation of A and B. So these two are like super important for the actual kind of communication. And you see that over like a very wide range of these two parameters, the color corresponds to the variation in the location of the boundary. You see, it's significantly sort of low. I mean, reduced compared to what you would observe in the case where there's no coupling. For these two cases, basically where S inhibits B or S upregulates A, right? For the other two, it's not so clear. But it's kind of not surprising that these two are doing well, because it's not surprising that this is doing well, given that this is doing well, because S inhibits B means that S is sort of indirectly upregulating A, right? So these two are actually kind of similar in that way. Okay, I'll just move out of time, so maybe I'll just to mention that one can also do an analysis by just looking at pairs of cell along the domain, right? So even if you just take a pair of cells and you just look at how the time series profile changes, you see that in the uncoupled case you have this sort of overlapping, and they sort of get nicely separated when you allow for S to inhibit B. Okay. This itself might take a long time, so I won't explain this. This is, but this is just to mention that one can also sort of do an analytical kind of investigation into the change in the noise levels. Basically, by following what's called the linear noise approximation approach, one can actually sort of analytically find the covariance matrix, and from the covariance matrix you can look at the variance in the quantities A, B, etc. Over space, and what we find is that once again we get something like which is inconsistent with our results. So basically, at the location of the interface, you see like sort of a high level of noise, sort of high level noise at the interface, and then that's sort of reduced once you have S inhibit B, and this also occurs even when S upregulates A. But for those of you who want more details about this, I'll mention on the final slide that so we have a mathematical notebook of this which goes into all of the details. So if you want, you can check that out. Okay, so this is the last result. So basically, this is just to mention that. So up to now, I haven't talked about the noise level. Right? So just taken a fixed noise level, but one can also really crank up the noise level to see how does that affect the result. And what we see here is that actually over like a couple of orders of magnitude, the result doesn't really change all that much, right? So going from say 0.001 to 0.1, the results are almost the same. After a point, you can see it begins to break down a bit, but nevertheless, it does really well over a couple of orders of magnitude. Okay, fine. So that's what I want to say. So yeah, so basically, what these results seem to suggest is that the notch signaling could play a role in enhancing the precision and the robustness of these cell fate boundaries. by utilizing the sort of the inherent asymmetry in the response of these genes to the morphogen. And also, I mean, this is something which could also potentially be experimentally verified. Like, so if you if you have a scenario where notch signaling is more effective at reducing so what we're hoping for is that you have a scenario where notch signaling is more effective at reducing noise when it upregulates the patterning gene, which is expressed at low morphogen concentrations. So so that's consistent with what we were saying. So this is something that potentially we can look into in the experimental literature of experiments have been done. We can look into that. So so this is the paper, and the codes are all over here on this site. You can ask me later if you like them. And so to stop. So I'd like to thank my collaborators once again. You will be hearing from Chandrasekhar next week. He'll be giving a couple of talks. And that's it. Thank you. And let's thank you in semaphore.
I have actually two questions. First one concerning the first part. Probably it's a quick one. Like you had this for this type 4 very alternating patterns. Is it like one by one? Yeah. Is it something? How you get this one by one alternating patterns? Is it just this example, or is it something hidden there that lets you saying like my first thought is like something like self-emerging, like maybe not like Turing pattern, but close to that. Saying what would be the mechanism for that? And
I mean, it's something that actually I mean, it's not too rare. It does happen quite a few times. So we haven't really looked into what the mechanism could be, right? So this is happening in this scenario over here, right? So basically, when you have like sort of mutual upregulation. But yeah, I mean, we haven't gone into the details of this part information.
Okay. And the second question was for the second part of the talk. You partly answered it in the conclusion that this mechanism of notch kind of refining the boundary works best at the low expression of when the morphogen concentrations are low. But is it also if you would take high concentration, or if you move with this boundary, having the exponential gradient to different regions, how this performance would be affected by simply having higher morphogen in input?
So okay, I don't think I have a big picture of that. Okay, so you're saying that what if basically you shifted the morphogen concentration such that the boundary was sort of shifting towards like higher values like this?
Yes, something like this, or even to lower values. Saying like, is there anything like that would I don't know. Is the model providing you the same effectiveness in two? Like minimizing the noise irrespective of where you are in the gradient, or or there is a dependence?
Well, okay, no, so right, no, so so we have varied the parameters related to the gradient itself. So these are basically this alpha m and so on, and tau m. So with that, you can tune like where the location of the boundary is, so that's just going to shift basically this to the left and right slightly. As far as we recall, that did not change the outcome all that much. But actually, but as you were mentioning this, it's sort of something else that I didn't actually mention when I was talking, was that if you notice here, the one thing that happens is that the boundary is happening like roughly around 20, right? In the case where there's no coupling. But when there is coupling, so if you notice here, you see that the boundary itself is sort of shifted towards the side slightly, right?
Okay.
Yeah.
So just because you have coupling, then kind of okay. So that's
So this is another thing that we were trying to explore. Like is there like the origins for this sort of shift, and is there some way to sort of to kind of have a kind of counter mechanism to sort of to bring it back to where it was originally. So it's sort of enhancing the robustness by sort of pushing it like closer towards the source in a way. The boundary pushing the boundary closer towards the source is sort of effectively enhancing its robustness. But yeah, this is it is an sort of interesting outcome over here, which we're still trying to sort of to untangle.
Yeah.